Thanks, Dinesh. So first of all, your dates are coming up pretty quickly. I'm sure you're all pretty aware of that. So I think your progress report is due next week. Now, given that next week's also Easter, some of your supervisors might be planning to take some extra time off. So just make sure that you speak to them in advance if you haven't already nailed down a time to go through your progress report. If possible, try and merge those different pages, scan those pages, and then attach them all as a single document. That makes our lives enormously easier. But use it as it's a really good opportunity to have a really honest conversation with your supervisor. Hopefully they will let you know how they think you're going. Often, I think at this stage, students are sometimes feeling a little bit unsure and insecure, and the supervisors will often provide a lot of reassuring advice at that time. And particularly, I think those students who are doing a fundamental science project at this stage, or probably up until now, are often going, oh my God, why did I decide to do fundamental science? This is so much harder than I expected. So, use it as an opportunity to have an honest conversation with your supervisor. Use it as an opportunity to let them know how you're feeling as well. We're used to students saying that they're insecure and they feel like they don't necessarily know everything, and or it might be that you say, can we meet more regularly? I'd really like to meet more regularly. I'm really struggling with this particular concept. So use it as a time to have a really good, honest conversation. Okay, so your literature review is due, I think, in 4 weeks. Yeah. Okay. So coming around fairly quickly as well. Okay, the most fundamental thing is to know that deadline. Exactly, because your supervisor isn't going to remember it. You need to remember it. Yep, and make sure that you get drafts of the literature to them well in advance so that they can read. Hopefully you've already started shooting parts of your literature to your supervisor to read. Look at the marking criteria. Yep, that's what you're going to get marked on. So read that rubric and look at the response sheet that they're going to fill in and make sure that you're addressing the criteria that you're going to be marked on. And be prepared well in advance. It takes so much longer than you expect, I think, writing the literature review. And the revisions particularly, if you've got a supervisor gives you, apparently there are some supervisors who don't read these things at all, a literature review and thesis. I'd probably like to hear get a confidential email if that's happening, if supervisors don't read any drafts at all. Perhaps not if you only send it to them the night before it's due, that might be fair enough. But otherwise, hopefully your supervisors will give you feedback on at least one good draft, and with any luck on two drafts of the thesis, of the lit review. Okay, so it takes time to give good feedback. Takes a lot of time. So really, I can't, say that enough times. Give your supervisor time to read drafts of the literature review. And I know lots of you are perfectionists. You don't like to send it till it's all perfect, but we're going to tear it to shreds anyway. So don't worry about that. Yeah, it will likely come back with lots and lots of either scribbles on a hand drawing, on a printed copy, or track changes on an electronic copy. So don't agonise over particular sections. If you're really struggling with a section, just put a comment in there. I really can't feel like I'm getting this section right, and your supervisors will help you. Discuss timing with your supervisor. If you haven't already nailed down time, particularly, this year was really terrible timing with Easter and Anzac Day and school holidays, all really close together with your deadlines. So make sure that you know whether or not they're going to be around and whether they're going to be happy to read drafts while they're on annual leave, if they're going away. And test your conversion to a PDF. Okay, so images just like to fly around documents all over the place. Incredibly frustrating, happens a lot. Make sure you've tested that conversion to a PDF before longer than 5 minutes before the deadline. Yeah. Sometimes, particularly if you've got a Mac, they're often not very good at shrinking your PDFs, so sometimes the PDFs end up massive. There's lots of really cheap apps that you can buy to help reduce the PDF size. Anybody writing a systematic review for the literature review? Yeah, just one? Couple? Okay, so if you do, hopefully you've seen the resources on Moodle on how to write a systematic review. One of the things to be really conscious about is reading those instructions, the marking guidelines, and make sure that your systematic review has to lead logically into your aims, hypothesis, and proposed research methodology. So really make sure that that's very clear. It might be that your systematic review is only covering a portion of the background literature review, so you might need to add some additional information beforehand and additional information afterwards to make that a logical story up to your aim and literature review, aim and hypothesis or research question. I'm just going to talk about a standard literature review today, not the systematic reviews. Remember, we're pretty tough on extensions. The faculty has been audited on special consideration, and we are really strict. If you've been sick for a few days before the deadline, that is not enough for an extension. It needs to be really serious, hospitalised, either you or a family member or partner perhaps hospitalised, something really serious in order to get an extension. Really, if you haven't already done so, or if there's sections that are missing in your literature review so far, just leave them as dot points as the headings and make sure you discuss that with your supervisor. You'll probably find, if they're anything like me, I'll go, oh, yep, that dot point outline looks great. And then Shag will attest. You send a draft like, oh, no, move that, put that section up there and that one down there and that one up there. You know, we'll do that. And then the next time you send it, I'll go, oh, no, move it back the way it was. You know, it just depends on the flow on those linking sections, okay? So that's pretty normal. When we send drafts of things to each other to read, drafts of papers, we do the same to each other. That's normal. And give yourself a timeline, particularly if there's only 4 weeks left now. Make sure that you absolutely have a plan of what you're going to achieve in each of those 4 weeks and exactly when you're going to get those drafts to your supervisor. Okay, so your literature review organisation. I always talk about the literature review as taking somebody by the hand and leading them along a garden path. So they need to know right from the outset where you're going to take them, and you're going to guide them every step along that path. Okay, it's really important. You start at the absolute basic fundamental concepts that the assessor needs to read. You don't assume background knowledge. You can assume that the person reading it is going to be relatively intelligent. They'll either have a science or a medical background, but one of them certainly is unlikely to be an expert in the field. So you really need to take that into account and start from the absolute fundamental concepts and then gradually build to the more complex areas. And it should be absolutely obvious what your aim and hypothesis are going to be before you get there. It should actually be obvious from the very first paragraph of the literature review. Language needs to be accessible for non-experts and use figures to highlight important concepts. Figures really are worth 1,000 words. They really are, particularly if one of your assessors is not an expert in the field. Headings and subheadings, can't say how important these are, because often you'll get distracted. You know, it's pretty rare that you actually get to read a document from start to finish without getting interrupted. So at some stage, it's likely your supervisor, the examiners, will put it down and come back to it later. So it's really much easier for them to find where they're up to and recall what they've read so far if they can just quickly flick through those headings and subheadings. Provide clear links. So the last sentence of each paragraph should really provide a logical link to the next paragraph or to the next section. Make sure you keep it relevant, focused and concise. So later I'll talk about the word limit. Lots of students will go like, oh, I have to get it exactly 7,500 words. I can tell you as an examiner, we'd far prefer that it was less than 7,500 words. Yeah, we'd far prefer to read something that's really concise and good than something that's just voluminous words. So this is generally how we think about the structure of any literature review. We put it in this sort of inverse triangle. We start here with really broad concepts, and gradually, as we walk along the path, we narrow the focus of the literature review. So it really should be starting from big, wide, fundamental concepts and gradually narrowing down to the specific area of your research project. So this introductory paragraph, I'll show you in a minute, should actually, that one paragraph should contain each of the major points that the assessor will need to know in order to understand your aim and hypothesis. So you'll have about 6 sentences usually, and that will basically be the major headings of your literature review, and already they will know what the aim of the project is going to be. You start with, now I've got some arrows here. These aren't always in, this is a broad sort of concept of how the structure will look. You often start with the description and scale of the clinical problem and the broad background to the area. Then you start to narrow it down from that broad field to a specific area. You talk about, now you're starting to get down to more detail. So you're talking about what's known and not known. You might talk about the potential causes and then outline the evidence for your particular project. Yeah, what's the evidence that suggests that your aim and hypothesis are a reasonable aim and hypothesis? And then, of course, finish down here. Now, as you'll see probably in the example that I'm going to show you, we don't always do it in, so some of these areas we might swap the order so that we've got a logical, just if the links are easier to make. So this is not set in stone way to write a literature review, but generally this tailoring it down, narrowing in an inverted triangle is the way that most literature reviews work. So this is an introductory paragraph. I'll just give you of a student from quite a few years ago now. I'll just give you a minute to read that. So, and from this paragraph, you should be able to essentially tell what the aim of the project, what the aim of his honours project was. Yep, so what do you think the aim was going to be? Yeah, that's right. To look at one potential mechanism that might contribute to bronchopulmonary dysplasia, and in particular, this protein called connective tissue growth factor. Yep, so it's not word for word the aim, but it's broadly, the assessor immediately knows, okay, we're talking about premature babies, we're talking about a disease called bronchopulmonary dysplasia, we're talking about a potential mechanism, ventilation-induced lung injury, and now we're talking about a potential mediator of ventilation-induced lung injury. So it's a really logical, they already know what the end of the path is going to look like. So how does that relate to the inverse triangle? So you can see that the clinical problem has been raised here. The broad background, so the start of, if I have a student who's writing a project in this broad area, they will nearly always start off by talking about the stages of lung development, the 5 basic stages of mammalian lung development. Now we're starting to talk about potential causes, assisted ventilation and lung injury. So what's known? Now we're talking about what's not known. We're talking about potential causes or mechanisms. What's the evidence to suggest that these factors might be involved, and what's the hypothesis and aim going to be? Yeah? So you can see it didn't follow the exact order that I had before, but essentially all those elements are there. And then often it's a good also to bring it back out to the big picture again in your final sentence. So what's the ultimate, what's the big picture reason that I'm doing this project for? Okay, so basically, and as that very first paragraph that you put up there essentially gives you the major headings of the literature review. Yep, they were the most important things that you need to get across to your assessor in order to understand the literature review. So immediately, just in that first paragraph, you can tell what the main headings were going to be. Now that triangle reverses when you're talking about the degree of detail. So you started with the big picture and you narrowed it down to a specific topic, but the amount of detail in each of those areas increases as you narrow it down. So the greatest detail that you'll have are in these final sections of the thesis. That's where you're quoting all individual research articles, and you might go into much more detail about the methodology that they happen to use to really support what the evidence is for and against your particular aim. Okay, so as you progress down that inverted triangle, you get more and more detail in each of those sections. Just a few fundamentals. Try not to use too many abbreviations. You've probably already, as you've gone around rotations in the hospital, learnt that the same acronym doesn't always mean the same thing when you're in a different unit. So that can be really confusing. So I give lectures on gut development and I talk about AECs, which are apical endocytic complexes, and I talk about alveolar epithelial cells in the lungs. It's the same acronym. They're both AECs. So, but I'm primarily a lung person. So if I see AEC, I'm always going to think somebody's talking about an alveolar epithelial cell. Yep, so remember that your assessors might be from a completely different field, particularly if you're writing anybody doing a really hardcore signal transduction project, cancer biology or, yeah. So you're going to have thousands of acronyms, but it might go to an assessor who isn't used to understanding signal transduction pathways. So you really use figures and diagrams to your advantage there, and only use the acronym if you really need it. You should only need to use an acronym if you're using it more than about 5 times. If you're only mentioning it once, don't bother putting in the acronym. Put a list, it always helps your assessor if you put a list of abbreviations in the front. Most of the types of research that you guys are going don't use footnotes. If there's people doing bioethics projects, it's more standard to use footnotes in that field. But most of the fundamental science or clinical project don't require footnotes. You might want to include a glossary. I think it's pretty rare that students do that, but sometimes they do. Now, as a scientist, we read every sentence. You probably already have realised from some of your supervisors, we can be total grammar Nazis, we can get really stuck on sentences. Do you really mean to say this? And you go, well, of course I didn't mean to say that. I didn't say that. And then when you reread the sentence, you realise that it could be interpreted in a different way. Okay, so science, medical research and science is all about being accurate. Yep, so every single sentence has to be absolutely precise about what you're talking about. Avoid these long convoluted sentences. You've lost track about what you were talking about. If your sentence is 4 lines long, it's too long. 3.5 lines is too long. So try to keep it less than 3 lines long for any sentence. Avoid jargon. So one of the typical sorts, if you're doing scientific writing, so you need to use precise scientific words. So, in the Ritchie Centre here, we do a lot of fetal sheep surgeries. And somewhere along the way, we got in the habit of calling the person who peels the surgical items for the surgeons, we got in the habit of calling them the peeler. And every now and again, you read these honours theses that say the peeler does this. Now, to somebody else, it's completely meaningless. Yeah, it's the theatre assistant, okay, or the scrub nurse or something. But be precise. Try to avoid those jargon terminology that don't make sense to anybody else. Avoid ambiguous statements. So read this sentence and tell me what's ambiguous about it. What's ambiguous about that sentence?
There's 2 very different ones to describe, isn't it?
Yeah, so this sentence here This sentence here says that study suggests blah, blah, blah. But actually there's two studies. Here's one study that showed something, and here's another study that showed something. So the next sentence doesn't tell you which of those two things you're referring to. This is what I'm talking about, accuracy and preciseness. You really need to clarify. Every sentence needs to be absolutely clear to your assessor exactly what it is that you're trying to tell them. I'm reading drafts of honest theses at the moment as well, of course. So this is an absolutely classic example of things that examiners pick up on. So this sentence, the function of blah, blah, blah is to do this, this, and then got some other roles. Yeah, now, as an examiner, we go, well, what are those other roles? Why aren't you telling me what those other roles are? I want to know what those other roles are. I'm going to ask you that. I'm going to mark you down because you didn't tell me what those other roles are. Yeah? If you're giving an oral presentation and you say something like that, immediately, that's a red light to somebody to ask you a question. Sometimes you do actually leave on purpose, leave a hole so that somebody will ask you a question, but don't do it. Definitely don't do it in your literature review. It's like a red flag to a bull. Okay, so if you have something like that, sometimes you don't want to list those other roles because there's hardly any evidence or it's a bit distracting, it's taking you off topic. So the way to get around that is to say something like the main functions of that is to do blah, blah, blah. Then you don't need to bother about listing these things that aren't main functions. You've clarified you're only talking about the main functions. Or you might say, to put them off asking you that question, you might say the main functions are this. Several other roles have also been posed, but there's currently limited evidence to support them. And then you just provide a reference. So you've sort of discounted those things. They're minor things. I don't want you to think about those because I want you to keep on track about the area that I'm focused on. Yeah, so there's ways that you can get around. Spell check. Please do a spell check. It's really annoying as an examiner to get something that's full of spelling and grammatical errors. That immediately, we have a separate part, I think, in the assessment sheet about spelling and grammar. But if it's full of spelling and grammar, you'll get marked down in every section, because it's distracting them from the main point of what you're trying to tell them. Not everything gets picked up in your spell check. So presumably most of you aren't working on ancient Greek artifacts, but you might be talking about experimental artifacts. Yeah. So again, just check your spelling. Clarify if you're doing something for, again, those people who work on signal transduction. If you have to talk about genes and proteins as part of your project, then you need to explain things to people who are not familiar with that area. So in most species, we have a particular nomenclature where you use capital letters for the protein and small letters when you're talking about the gene. So the first time you say that, you need to actually spell it out absolutely clearly, or the assessor is going to think that you're making a mistake, you've been completely inconsistent throughout your thesis. Yeah, so if you've got something like that, make it really clear and stick it in your abbreviation list. Track changes. Whose supervisors have given them feedback using track changes so far? Yeah. A few, right? It's a really easy habit to just go, click, accept, 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 accept. Oh my God, they've left a comment. I actually have to think about it. Yeah, don't just blindly accept those track changes. Really think, why have they suggested that change? Was it because it was ambiguous? Was it because the sentence was too long and convoluted? The only way that you will learn how to write scientifically is by really paying attention to what your supervisors have suggested. Now, sometimes it's our little idiosyncrasies. We just have little preferred ways of doing things, but most of the time there'll be a clear reason why they've suggested those changes. There is some online material to help identify common sort of mistakes. Referencing, you often get lots of questions around referencing. Reference, usually in that first paragraph, you wouldn't have any references. It's a broad overview. You're going to provide all the detail later on. So the first paragraph you wouldn't normally reference. Thereafter, you need to reference every specific statement. Okay? Reviews are okay for broad statements. Those first few sections of your literature review, often you're referring to things that have been known for 20, 50 years, and you know, you might not even be able to track down the original papers of who found them. You can just refer to a review article. Try to avoid textbooks. Textbooks are not very up to date. Textbooks are terribly out of date. So try not to refer to textbooks. Far better to refer to a review article. When you get down to the very end, those final parts of the literature review where you're going into much more detail, we won't expect to see any reviews in there. Yeah, because we're really talking about individual studies and the evidence in those individual studies that suggests that your aim and hypothesis are valid. You will be amazed. Again, an easy thing when you're really busy is somebody, they cite another paper that said this, and you believe it. Don't believe that, because you'll be amazed at how many times people miscite other papers. You'll be amazed at how many times people miscite themselves. Yeah, you'll read an abstract and go, fantastic, that supports my hypothesis. Then you go through and you read the methods and you read the results and you go, hang on, they've just said the exact opposite thing in the abstract than they actually have shown in the results. Yeah, it happens. Okay, so you really do need to go back and check the original source of the data. EndNote, is everybody using EndNote or some other program? If you're not, you need to do it right now, really soon, because it takes a long time. Similarly, set up your headings using table of contents as you go along. If you don't know how to do that, I'm sure there's easy YouTube clips or one of the other students will be able to show you. It will save you an enormous amount of time to generate your table of contents at the end, particularly when you have a supervisor like me who makes you change sections around. Okay, don't hide behind discrepancies in the literature. You know, you've come out of medicine where things are black and white. We do this because of this. We do this, we just do this. This is what causes this. Now you're entering the realm of the unknown. Yep, so there will be discrepancies. There will be one study that shows something increases and another study that shows it decreases. You need to highlight that. Don't shy away from showing that some studies show that one thing happens and other studies show that things don't happen, because that's the difference. That's what happens with experimental design. It can have a big impact on the outcomes that you measure. And depending on where you are in that inverted triangle, you might discuss the reasons for those discrepancies. So if you write down here, in the pointy end of the triangle, then you might talk about why it is that that particular paper showed an increase while that other particular paper showed a decrease. Figures. Be really generous with figures. Examiners, we love figures. It helps us understand things, helps us remember things if we're not an expert in the area. If you are using figures then you must reference the original source. And if you're using the figure legend identically from the paper, then you need to have that in quotation marks. Yeah? Otherwise it would be picked up as a in Turnitin, as an episode of plagiarism. Okay, so if you take a figure directly from another article, then you state taken from. If you modify a diagram, then you say modified from. Yep, it's okay to modify other people's diagrams. Often they've got 100 extra labels that you don't want to have on the figure. You just want the most relevant diagrams. So this is what a figure should look like in your thesis. It should have a figure number, should have a figure title. Normally we'll put them in bold. For a figure, it's usually underneath the figure, and then there should be a figure legend that explains it in detail. If there are things that aren't labelled, like up the top here, then it should be clear that that's, in this example, is referring to weeks. Yep. So if there's an acronym or something in there, then you need to explain what the acronym is. How many references? People like to know an exact number of references. This is going to vary enormously depending on the field and how much is known in that field. I think if you're doing more than 120 references, you're probably going a little bit overboard. We don't like to read a sentence with 10 references at the end of it. We're normally pretty convinced by 1 or 2. I think most theses will have more than 50, but if you're working in an area that's really new, it might be that hardly anybody has published in that field yet. This year, for the first time, our word limit is hard. 7,500 words, no more. Yeah? So that's because when we as scientists, we don't have the flexibility of plus minus word limits. When we submit an NHMRC research grant for our next 3 or 5 years of funding, we have 9 pages, but 9 pages including references to describe all of our findings, whether it's a 3-year grant or a 5-year grant. Yep, no flexibility. When you submit a small application to a philanthropic funding body, then they'll say 150 words for this section, a maximum of 2,000 words or characters in this section. Yep, so you need to get used to having very hard limits. So no more than 7,500. That's already a lot of words anyway. The limit excludes fundamental things. The limit excludes cover page and declaration, etc. You can read that in the unit guide. Your headings and your in-text citations are included in the word count. So it's best to use, if you're using EndNote or something, then choose a journal style where it has numbers rather than Wallace, Narula, 2, XYZ, somebody else, you know, that's going to take up a lot of space. Choose one. Numbers is usually the best way to keep your word limit within the right frame. Turnitin, another major change this year. The university has decreed that if you're past second year of university, you will no longer get to see the matches from your Turnitin reports. Okay, so they figured that you've already been through, everybody's seen used Turnitin before? Yep. And you used to be able to submit to Turnitin and then you got to see where the matches were? Yep. So you haven't done that before? Yeah. So they figured that by the time you're at 3rd, 4th, 5th year, that you should already know how to write sentences in your own words. Yeah. So the easy way to get around that is never write identically from a journal article. You can have a separate document where you might copy paste sections of text, but in your literature review, don't ever write anything. It goes from that document, everything in your own words in your literature review. There are some things that come up in honours thesis that I see as matches all the time. I'm not worried about. So the table of contents. So if you were doing, most students, honours students in my lab would have a table of contents that the headings and subheadings would be really similar, at least the first half. Okay, so that would come up as a match. That always comes up as a match. I don't care if your table of contents is nearly word for word the same as some other student from your lab. I don't care about that. Definitions. There's only so many ways you can define something without changing the definition. Yeah, so that nearly always comes up with a match with somebody, some piece of text somewhere. If you're defining hypertension, for example, you know, there's only so many different ways that you can write it. So if you want to really, I don't care if I see something that's clearly a definition. I'm not too fussed about a fairly high match rate in that portion. But if you're worried about it, just put it in quotation marks. Take it directly from, you know, whatever the clinical guidelines are, and use that. Put it in quotation marks. Questionnaires. If in your methods part at the end you're attaching a questionnaire, that questionnaire has been used by some other student, of course it's going to be a match. Yeah, I don't care about that. That's logical. If you're using methodology that's identical, a piece of methodology that's identical to somebody else, I'm not too worried about it either. And ethics statements always come up as a high match as well. So this is what I would see in lots of people's literature reviews, a sort of match. Obviously I couldn't take somebody's actual Turnitin match. So I've pretended, using that same paragraph as before, I've pretended the sorts of match. This is a sort of match where it's quite clear that students attempted to write it in their own words. Yeah, I'm not too worried about that. If it looks like this, however, I'm going to be worried. There's whole sentences in there that are word for word, or a sentence where maybe there's only one word that's been changed. So those are the things I don't like to see. So I indicated this at the start. One of your examiners will be nominated by your supervisor. It's likely they'll be an expert in the field. They'll know a lot about your field. The second examiner, we nominate from within the pool of other supervisors. So they're unlikely to be an expert in the field. It might be, by chance, that they happen to know a lot about your particular organ or tissue or disease, but chances are they may not be an expert at all. So you need to take that into account. Remember that lots of the supervisors are scientists, not clinicians, and so you just got to remember to tailor it to your audience, to a broad audience. Okay, and your supervisor, of course, will be asked to examine the literature review from another thesis, from a literature review from another student. Okay, and that's usually the same. In 99% of the cases, the two people who examine your literature review will be the two people who examine your final thesis. So pay attention to their comments when they come back. Oral presentation, I'll just really quickly go on oral presentation. So it's 10-minute talk and 5 minutes of questions. That usually means 10 to 15 slides. Again, check the marking guidelines. What are you actually being marked on? Make sure that you know and you've prepared your presentation accordingly. In an oral presentation, we only expect you to reference really important points. You don't need to reference every single sentence there. It's not like your literature review. Less is more. You often have students in a literature review, giving their oral presentation, and they try to get everything in, and they're going through their slides, and they're doing this, and you can't hear them. You don't have time to read the slide. You can't understand what they're saying. So less is always more. Keep your slides really simple. I have failed that in many of the slides in this talk. Every time I've put up a paragraph like something like this, I mean, you would never normally put up something like that in an oral presentation because you just can't. You wouldn't expect your audience to read it. Again, you should be taking the audience by the hand. It should be a really simple, easy story across only 10 or 15 slides to explain where the basic fundamentals, what's the big clinical problem that you're addressing, and then what's taking them to your aim and hypothesis. So keep it really simple. Try not to read. Yep. Really try not to read from notes. Much better. If you're a really nervous speaker and you like that security of having notes, just try and have logical prompts on your slide. That's usually all you need. But the prompts need to be meaningful to the audience as well, not just to you. So the word prompt on its own is meaningless, but if you have used prompts as guides, then it makes sense to the audience. Yeah. And good links, again, really important. The final sentence of each slide should logically take you to the next slide. Diagrams help. Shag's going to recognise this one, particularly when you get to your methodology. Timelines and experimental interventions really helps if you include detail. We generally break it up into about 60% background and about 40% your proposed methodology. Really important if you're doing stats that you mention the stats that you're going to use. And try to finish with an expected outcome slide. So bring it back out. What's the big picture question? What's the reason that you're doing this study for? And prepare backup slides. We don't need a slide with references on. Don't bother doing that. Common mistakes. These are the sort of really common mistakes I get. I'm really nervous and talk really fast, and nobody can understand me. That's the most common. So when you come up to the stand away from the little microscope for a minute, and just take big breath and relax. Yeah, slow your heartbeat. The other thing is that people are really nervous and they speak really quietly and then they can't hear. Yeah. So you need to use volume. Use the volume, speak slowly, speak clearly. Try not to speak, try to get some intonation. You'll see some people, I'm a probably excessive intonation in the way that I speak. I'm certainly an excessive hand user. That can be quite distracting. If I was giving a formal presentation, if I wasn't talking to students and I was at a conference giving a formal presentation, I wouldn't be doing this quite as much. I'd be much more serious about my presentation. When pointing, so if you do happen to have a lot of detail on the slide, it can be, or it's a complex slide, it can be really difficult for people to follow. So really take advantage of having a pointer to highlight what it is that you're interested in. It's okay to look away. If you're pointing, you obviously need to look at the slide, but otherwise you should be engaging with the audience. People, if there's a lot of seminars going on in your slide at 7 o'clock at night, just after lunch, everybody's got that postprandial nap thing going on. Look at them in the eye. Yeah? I tell you what, it really, I go, ooh, better pay attention. Okay, so really make sure that you scan the audience and make eye contact with those people, because then they will pay much more attention to your presentation. If they're paying attention to your presentation, they'll understand it and they'll give you a higher mark. Distracting, as I said, distracting, I go like this. Usually that's because I'm giving lectures to students. I'm like trying to keep them awake. But, you know, avoid other distractions. You know, it's fun when you first start playing with PowerPoint. You get all these animations and you can do cool stuff and you can put in really funny things on your slides and you go, that's really cool. But actually, in a scientific presentation, that can be really distracting. One of the probably best examples, I told these guys last year, when I was in the Department of Physiology and we had an honours student who was working on sperm. And he had a slide, and the fundamental background to the slide had a built-in animation. There was a little white thing from here and it would zoom across the, spread out and zoom across the slide. The moment you flipped to the slide, it looked like every slide was ejaculating. So at every slide, the entire audience was crying. By the end of the presentation, there were tears running down people's faces. They were laughing, and then they go, oh, we've got to concentrate. You could see the laughter would dim, and then somebody would start giggling, and the entire audience would erupt in laughter again. It really was, it's the funniest talk I've ever been to. But nobody heard what he was talking about. You couldn't mark him because we didn't actually know what he was talking about, because we couldn't hear him over the laughter. So just, you know, bear your animations in mind. Use them to your advantage, not to your disadvantage. The other thing is try to speak conversationally. Often if you're nervous, you'll try and rehearse word for word. You know, what you've written down, your pre-prepared thing, you want to keep exactly the time, so I need to say exactly the words I've pre-prepared. That's an absolute major way to completely lose the words that you're looking for when you're giving the presentation. Yes, so try and make it a bit more conversational. We want it to be a conversation with the audience. Also be careful using written terminology. So this is something that makes, we go to Japan twice a year to do experiments, and every year we have to sit through this radiation safety training, and we laugh in it because they use words like, moreover, when you do this. It's like, moreover, nobody ever says that when you're speaking. You might write it, but you never say that when you're speaking. Yeah, so just remember that this is an oral presentation. Use spoken words, not written words. Practice out loud. It can be word perfect in your head when you're saying it silently. It's very different when you need to say it out loud. When it comes to question time is absolutely the hardest part of giving an oral presentation. Absolutely the hardest and most scariest part. You have no idea what they're going to ask. It is okay. When you're a clinician and you want to give confidence to your patients and your fellow clinicians that you're working with, that you know everything. You're confident and you need to exude that confidence. But now you're doing scientists. We're working in the realm of I don't knows. There's lots of things that we don't know. This is when you're really starting to learn about all the I don't knows. So it's okay to say that. If somebody asks you a question, it's okay to say, that's a good question. I don't know. In fact, I'm not sure if anybody knows. Yeah? Or I don't know, I'll look it up. Or do you know? Throw it back to them, because I don't know the answer to that. Do you know the answer to that? Sometimes we ask it out of ignorance. We don't know what the answer is. And sometimes we're testing it. But it's okay, because then they get to spout their knowledge. They go, oh, I'm pretty good because I know the answer and you don't. Yeah? So I wouldn't do that too much. But usually it's okay. It is absolutely perfectly okay if somebody asks you a question to say, I don't know. It's far better to say, I don't know, than to say something that is wrong. Because if they know the answer, and particularly the first seminar, we're often testing your knowledge when we ask questions in that first seminar. So it's really bad. It is one way to lose a lot of marks if you say something that's wrong, if you just make it up on the spot. Far better to say, I don't know. And prepare backup slides. You can't stick everything. If you've got anything complex in there that you don't have time to explain in detail, then stick in the backup slide. And you click on, here's one I prepared earlier when they ask a question. Okay, it looks really good, looks really professional if you've got pre-prepared slides for some of those questions. You should be able to go through pretty much every single slide in your talk and think of a question that could be asked. Yeah? So really try and think in advance. Talk to your supervisors about what questions you might get asked. You'll only be able to predict a proportion of the questions anyway. Again, there's some online information to help. And the thing I've forgotten to put in there in the literature review, back up, back up, back up, back up, and back up again. Yeah, absolutely. That is absolutely when your files are going to crash. So make sure that you back up everything, back up regularly. You write a great paragraph you've been struggling with for ages, and all of a sudden, oh yes, the words are flying out, fingers are going at the speed of light. Save. Press that and save it, because you'll lose that train of thought in a minute. Okay, any questions? No? Yeah?
My supervisors sort of suggested that I do go through the project. So I'm not, like they link together, but I'm not sure.
Yeah, so this is, I mean, we often there might be more than one aim in a project, and that's perfectly okay, as long as they link logically, as long as the background covers it, and usually the one project links logically into the next project. If they're not linked, that's when you come up with problems. If there's no logical link between project one, or the literature review is clearly separated into half about aim one and half about aim two, and the two aims are not linked, then don't do it. Because when it comes to the discussion, you're only going to have half the amount of detail on each of those aims, and you'll get marked down because you haven't gone into enough detail.
So provided that the background sort of covers both of them, while in each way, I can still discuss both of them.
Yep. Just label them as hypothesis one and aim one, and hypothesis two and aim two, something like that. Anything else? No? Good. Okay, well, good luck. It's only a few weeks left. I live up on the 5th floor in this building, so, you know, we're emails and phone calls away. It's a pretty stressful time coming up to that last week of your literature review for most people, particularly given that we've got the Easter break and Anzac Day break so soon beforehand. Okay, so good luck, and I'm going to run away, leave you to these two to give you the honest truth.